Good afternoon and welcome to Sequencing Team Office Hours. For those that do not know me, my name is Stephanie Lunn, and I am a bioinformatician on the molecular epi team here at WaDOH. Previously in bacterial genomics, we started off with part 1 of bacterial genomics fundamentals, and we talked about some assumptions of bacterial genomes, how the genome was organized, and some of the content found in bacterial genomes. In part 2, we continued on with our theme of a bacterial genome as a concept. We distinguished between core, pan, and accessory genomes and discussed a little more about the assumptions of bacterial genomes. In today's session, we are continuing on with part 3, and we are going to be talking about some concepts that is essentially classical bacterial genetics. And a lot of these concepts presented in this lecture are by the book, so to speak, but I will try to keep it at a medium level detail so you can learn how these mechanisms work, but without getting too bogged down into technical detail. So let's start our journey by talking about the diversity of bacteria. The range or diversity of bacteria is greater than in any other life form. And this phylogenetic tree shows the phylogeny of the bacterial 16S rRNA sequence, which is a sequence belonging to part of a ribosome, and that is the site of protein synthesis inside of a cell. And if we look to the right in yellow, we see many different major bacterial phyla. Each of these groupings of bacteria live and survive in different habitats that far outweigh many organisms on this earth. For example, we have Chlamydia, whose members are quite diverse and include pathogens of many animals, including humans, and even some symbionts of protozoans or unicellular organisms. We can see Actinobacteria, whose members are very important to us because of their contribution to the soil systems. And we can see Spirochetes, which are corkscrew-shaped bacteria. And some members in this phylum are significant pathogens that cause severe disease like Treponema pallidum, the causative agent of syphilis. And to just mention a couple of other, there's Acidobacteria, whose members are quite physiologically diverse and can be found in a variety of environments. You can find them in places like soil, hot springs, and even the ocean. And lastly, we have Proteobacteria, which I think has recently been renamed to Pseudomonadota. Members of this phylum can cause disease like Yersinia, Salmonella, E. coli, and even Legionella, but not all of them are pathogenic. Some of them are free-living and even responsible for the important process of nitrogen fixation. So interestingly enough, the range of environments for bacteria is so diverse that even some of these bacteria coexist in a microcosm in our gut microbiome. So bacteria inhabit just about every niche of the earth, and you may be wondering, how is it that they are so diverse? The diversity of bacteria is due in part to higher levels of horizontal genetic transfer. Horizontal genetic transfer is the transfer of genetic information, specifically DNA, that can occur between unrelated bacterial cells or bacteria of different species. And it is important to note that DNA transfer in bacteria is unidirectional and always independent of reproduction or cell division. So this figure here illustrates the 3 mechanisms of horizontal gene transfer in bacteria. It includes conjugation, which is a type of bacterial sex because it requires cell-to-cell contact. There's transduction, a process whereby viruses called bacteriophages can insert their genetic material into a bacterial host. And there's transformation, in which bacteria can take in genetic material from the environment, say, for example, free-floating DNA from degraded or lysed bacterial cells. So as a result of DNA transfer, new combinations of genetic material that bacteria can acquire may be adaptive, such so that they can survive in different environments. And what this means is that bacteria can be genetically fluid and even functionally immortal. So with all that said, here are our goals for today. First, we're going to discuss the fate of DNA that enters the cell through horizontal gene transfer. Next, we're going to discuss the 2 general types of recombination and their impact on the genome. And recombination is very important as mechanisms of horizontal gene transfer can require recombination systems. After that, we're going to dive into the different mechanisms of horizontal gene transfer in bacteria, including transformation, transduction, and conjugation. And once we have covered the important concepts, we will connect this information with the concepts we covered previously on core genome, pangenome, and accessory genome. And lastly, all that I cover here today, I hope that it will set the context for how this knowledge can be beneficial to epidemiologic investigation and surveillance. So with an understanding of what I would like to cover today, these directly tie in with our learning objectives. There's just a few things you should be able to do after today's lecture. So first, be able to briefly describe the fate of DNA that enters the cell through horizontal gene transfer. Next, be able to define recombination and briefly describe the difference between non-homologous recombination and homologous recombination. And with this topic being centered on horizontal gene transfer, you should be able to briefly describe or at least define each of these three mechanisms. And lastly, be able to briefly explain how these mechanisms can influence the core genome, accessory genome, and pangenome. But first, before we get into discussing the mechanisms of horizontal gene transfer, we should first consider the fate of DNA that enters the cell through horizontal gene transfer. That is to say, what might happen to the DNA once it enters the cell. Whether DNA has been transferred by transformation, transduction, or conjugation, there are three possible fates. The first is that DNA may be degraded by restriction enzymes, as we see here in this part 1. And there could be several reasons for this, but one such reason could be a means of self-defense in the host cell. Bacteria contain modification systems to be able to recognize their own DNA to prevent it from being degraded by its own restriction enzymes. If foreign DNA that enters the bacterial cell lack this modification or even lack specific recognition sequences, then they may be degraded by the host cell's restriction enzymes. So this is one means of self-defense of bacterial cells against foreign DNA that could especially be harmful. The second fate of DNA that enters the cell through horizontal gene transfer is that it may replicate by itself. But this is only possible, though, if the DNA possesses its own origin of replication, which is often represented as ORI for short. Plasmids and phage genomes are two examples of DNA that contain origins of replication and are able to replicate by itself upon entering the host cell. And lastly, as we will talk about in the next slide, the last fate of DNA that enters the cell through horizontal gene transfer is that it may recombine with the host chromosome. When DNA enters the cell through horizontal gene transfer and recombines with the host chromosome, it can integrate new genes coding for proteins with specific functions or even generate new combinations of alleles. So let's talk about recombination next. Recombination is the breaking and joining of DNA and new combinations. It is the physical exchange of DNA between genetic elements or structures that carry genetic information. Recombination is often categorized as an evolutionary mechanism, and just about all living organisms possess mechanisms for recombination. Recombination introduces new genetic variation, heritable diversity that can be passed on in population. So for the purpose of this lecture, I distinguish between two general types of recombination, non-homologous recombination and homologous recombination. So non-homologous recombination is the recombination between specific sequences on DNA that occurs, even if the sequences are mostly dissimilar. In other words, the sequences do not have to share homology or be the same identical sequence in non-homologous recombination. This process can involve lateral gene transfer, which can occur between two unrelated cells. It includes transposition, which I'll talk about here in a moment, and that is the movement of certain DNA sequences. And it also includes site-specific recombination, which is recombination that occurs between defined sequences in DNA or at specific sites. And this is usually performed by proteins with this specific function, and they're called site-specific recombinases. Recombinases in general are enzymes that specifically recognize two sequences in DNA and joins the strands to cause a crossover within the sequences. Homologous recombination, on the other hand, is recombination that occurs between two DNA molecules that have the same sequence in the region of the crossover. We refer to these same sequences as homologous DNA sequences. Because the sequences are the same, bases can pair over an extended length of the DNA molecule. And as I'll mention more later, this type of recombination drives the process of crossing over in classical genetics. So I have drawn the primary difference here between non-homologous recombination and homologous recombination. And both processes generally work a little bit differently. And another way to distinguish between the two is this sort of conversion of the DNA sequence in the transformed cell after recombination has occurred. So let's start with non-homologous recombination. You can think of it as a form of lateral gene transfer. So say we have the DNA of a donor cell, which contains a series of genes represented by the green boxes. Since recombination requires the tango of two DNA molecules, we also have the DNA of the recipient cell, which contains a series of genes that are represented by the red circle. Note that the sequences between the donor cell and the recipient cell are dissimilar or not the same. Now, there are several different forms of non-homologous recombination, but for the purposes of illustrating this example here, we see that after recombination has occurred, a gene from the donor cell has been transferred to the recipient cell. And so the DNA of the transformed cell shows the new sequence with the transfer of this novel gene. Now, homologous recombination, on the other hand, we see that the sequence from the donor cell is similar to the sequence of the recipient cell. The sequence from the donor cell is represented by these blue circles rather than green boxes to represent homology. And between the two sequences, the circles could represent the same gene, but the color would represent different alleles, which are alternate forms of a gene. After recombination has occurred, we see a new combination of alleles in the DNA of the transformed cell. So to sum up this picture in one statement, a novel gene can be transferred from non-homologous donor DNA, whereas for homologous donor DNA, the new variation is limited to a new or even identical allele. So one example of a process that uses non-homologous recombination that I want to mention briefly is transposition. In the last two bacterial genomics fundamental lectures, I mentioned that one of the things you can find in a bacterial genome are transposons. And you might recall me mentioning them as these sort of jumping genes because they can hop from one place in DNA to another. Transposons are an example of mobile genetic elements, or MGEs, that move as DNA. And transposition is the process by which a transposon moves. I find them to be very important as transposons can carry genes for resistances to antibiotics and heavy metals. Transposons always carry recombinases flanked by these inverted palindromic DNA called insertion sequences. And palindromic DNA are sequences that are read the same backwards as forward, like so in this example shown here in this figure. And what this does is that this structure actually allows for integration into a chromosome target site without a homologous recombination, thereby inserting new genes or possibly even inactivating a gene or operon. Interestingly enough, transposons can also contain genes that encode the conjugation apparatus, which can make these transposons more mobile. The conjugation apparatus can include a hair-like appendage called a pilus, which can allow for bacteria to physically connect to each other. And transposons that contain genes encoding for a conjugation apparatus are called conjugative transposons or integrative conjugative elements, or ICE for short. The ability to code for their own pilus or transfer mechanism can allow them to move within cells and between cells. So in this form of non-homologous recombination, the sequence from the donor DNA does not have to be the same as the target DNA in order for the transposable element to get integrated into the target DNA. Homologous recombination, on the other hand, drives the process of crossing over in classical genetics, where two DNA molecules exchange genetic information, resulting in the shuffling of genetic information or a new combination of alleles. Homologous recombination can occur when closely related DNA sequences or homologous sequences from two distinct genetic elements are combined together into a single element. And during crossing over, the cross strands can uncross and then recross in a different orientation. So we see here in this figure, and I apologize, you won't be able to follow my cursor, but we see the donor DNA here at the top in green. And for homologous recombination to occur, the DNA is nicked at the specific site, which is represented by this gap. And then this allows for the binding of proteins to occur, including REC-A, which is actually key to homologous recombination. And this protein has been identified in just about all prokaryotic species that have been examined. And what it does is that it helps promote base pairing in the complementary sequence of the recipient DNA molecule. So what happens next is that the strand from the donor DNA will invade the target DNA, forming a cross-strand interaction where genetic information can be exchanged. Now, depending on the orientation of how the sites are cut, there are two outcomes that could occur. If the strands are cut horizontally, as represented by these yellow triangles, then they may result in patches. Whereas if the strands are cut vertically, represented by these blue triangles, then this may result in splices. And if you notice on the left, in the patch model, the exchange of genetic information is symmetrical. Whereas in the splice model, the exchange of genetic information is non-symmetrical, rather you see fragments. So moving on, we can actually talk about the mechanisms of horizontal gene transfer. And this slide summarizes each of the three mechanisms, but I'll cover them in a little bit more detail in the following slides. So the first mechanism of horizontal gene transfer that we'll talk about is transformation, and that is the transfer of genetic information via free DNA. The second mechanism is transduction, where the transfer of host genes from one cell by another by a virus to another by a virus. And so yes, even bacteria can become infected with viruses. And during transduction, bacteria can pick up new genes by accident. And then the third mechanism is conjugation, which is the transfer of genes from one prokaryotic cell to another by a mechanism involving cell-to-cell contact. But first, I want to talk about the discovery of transformation. And I won't spend too much time talking about experiments, but I do want to mention this one here, because this seems to be where our knowledge of horizontal gene transfer begins. Transformation was the first mechanism of bacterial gene exchange to be discovered. So as the story goes, in 1928, Frederick Griffith found that one form of the pathogenic Streptococcus pneumoniae, formerly pneumococci, could be mysteriously transformed into another form. As pneumonia makes two types of colonies with different appearances, one type made by pathogenic bacteria and the other type made by non-pathogenic bacteria that are incapable of causing infections. The colonies made by the pathogenic strains appear smooth on agar plates because the bacteria excrete a polysaccharide capsule, and this is called smooth colony formation. This capsule can protect the bacteria and allow bacteria to survive invertebrate hosts like mice, and when they infect these hosts, they can kill them. On the other hand, sometimes rough colony forming mutants that cannot make the capsule will arise from the smooth colony formers, and these mutants are non-pathogenic in mice. So in his experiment, Griffith mixed dead S. pneumoniae cells of the smooth colony formers with the live non-pathogenic cells, specifically the rough colony formers, and he injected the mixture into mice. So the mice that were given injections of only the rough colony forming bacteria survived, but mice that received the mixture of dead smooth colony formers and live rough colony formers died. Furthermore, Griffith isolated live smooth colony forming from bacteria from the blood of the dead mice and concluded that the dead pathogenic bacteria gave off this sort of transforming principle that changed the live non-pathogenic rough colony forming bacteria to live pathogenic smooth colony form. And he has speculated that this transforming principle was the polysaccharide itself. Now, other researchers have repeated a similar experience mixing rough forms with extracts of the smooth colony formers in vitro or in a test tube, and they found that their surviving cells included smooth colony formers. And this indicated that a subset of the active cells was present in the extract, so we didn't need mice for transformation. They weren't necessarily needed for transformation to occur. However, about 2 decades later, Oswald Avery and his collaborators purified this transforming principle from the extract of the smooth colony formers and showed that the active fraction of the cell's extract is actually DNA, demonstrating that it was DNA and not protein and not the polysaccharide. that is actually the hereditary material. And in the 1940s and 1950s, this was a groundbreaking revelation. And from these experiments, we learned that living bacterial cells can acquire DNA, the hereditary material, from dead cells if DNA was present in the environment. And this is one way in which non-pathogenic bacteria can become pathogenic. So in transformation, cells take up naked DNA, and a single cell usually incorporates only one or a few DNA fragments. So only really a small proportion of genes from one cell can be transferred to another in a single transformation event. And to put that into perspective, the Bacillus subtilis chromosome is about 4.2 megabase pairs. And when the cell is lysed, the chromosome is converted to fragments of about 10 kilobase pairs each. If the average gene is about 1,000 nucleotides long, then each fragment of the Bacillus subtilis DNA contains about 10 genes, and this is typical transformable size. And since a cell incorporates only one or a few DNA fragments at a time, only a relatively small amount of genes will be transferred in a single transformation event. Cells that are able to take up DNA and be transformed are said to be competent. But this capacity is genetically determined. And while certain species of both gram-negative and gram-positive bacteria are naturally transformable, high-efficiency natural transformation is actually rare among bacteria. Only 1% of the species are naturally transformable, but does include some important pathogens like Acinetobacter, Bacillus, Streptococcus, Haemophilus, and Neisseria. But some of these bacteria, like species of Haemophilus and Neisseria, can only take up DNA of only the same species. So if we look at this figure, we see the transforming DNA from the donor cell at the top here in purple. It's these purple strands, and they are binding to this orange surface protein of a competent cell. The recipient cell will take in that DNA, and what follows is REC-A mediated homologous recombination. And so in the transformed cell, the DNA becomes integrated into the chromosome. So moving on to transduction. Transduction is the transfer of host genes from one cell to another by a virus. And it can occur in a variety of bacteria, including Escherichia, Pseudomonas, Salmonella, Staphylococcus, among others. Now, there are two different types of transduction. There's generalized transduction, which I'll talk about here on this slide, and then there's specialized transduction, which I'll talk about next. So in generalized transduction, just about any gene on the donor chromosome can be transferred to the recipient. Bacteriophages involved in generalized transduction are usually virulent or lytic phages that could be comprised of RNA or DNA. And when the bacterial cell is infected with these phages, it causes destruction of the cell, and this releases new progeny phages. So I have here on this slide the process of the lytic cycle, whereby a phage inserts this genetic material into the host cell. And this genetic material is represented in orange. And so sometimes what will happen is that during the lytic infection, the enzymes that are responsible for packaging the viral DNA into the phage may also accidentally package host DNA in the process. And so what this does is that this results in a combination of normal phages, as you see here in orange, and phages that contain the donor cell DNA, which is called a transducing particle. So in generalized transduction, the recipient cell is infected by the transducing particle. Because the transducing particle cannot replicate, the donor genes will actually be lost unless they recombine with the recipient bacterial chromosome. So homologous recombination is required in order for DNA to integrate into the chromosome. Now, be that as it may, these events typically happen at a low frequency. And this is because, one, a portion of the host DNA is exchanged for phage DNA, which usually occurs by chance. And then two, most bacterial cells are typically infected with normal phages. So in contrast to generalized transduction, where the recipient cell is infected with the phage-carrying donor DNA, and specialized transduction, DNA from a specific region of the host chromosome is integrated directly into the virus genome, replacing some of the virus genes. And this is caused by bacterial phages whose DNA can integrate into the bacterial chromosome at specific sites using recombinases. Whereas lytic phages are involved in generalized transductions, bacterial phages involved in specialized transductions are typically temperate or lysogenic phages. And lysogenic phages come from latent virus infections where the phage genome replicates, usually integrated into the chromosome. So as shown in this figure, we see a part of the phage genome that has been integrated at a specific site into the chromosome, and this is called a prophage, and this is represented here in dark green. Also note the galactose gene present in the chromosome as well in pink. So in normal events, if we follow to the left, the phage DNA will circularize and then detach from the host DNA. And the detached DNA can replicate and form progeny phages, which ultimately result in the lysis of the bacterial cell to release the progeny phages. But in a more rare event, as we see here on the right, a portion of the host DNA is exchanged for phage DNA. So when the DNA replicates, it includes the host DNA. So in this scenario, we see the galactose gene here in pink. I know it's probably a little bit difficult to see, but we see that the galactose gene has been exchanged for phage DNA. The DNA replicates, form progeny phages, and this can result in defective phages that can transduce galactose genes. So even though this tends to be a more rare event, this can result in a more efficient transfer compared to generalized transduction. Before we talk about conjugation, we should cover plasmids very briefly. So bacterial cells can often contain plasmids in addition to the chromosome, and plasmids can play an important role in bacterial adaptation and evolution. Plasmids tend to be stably maintained in bacterial cells at low copy number per cell, about 2 to 8, or high copy number per cell, about 100 to 1,000. Now, generally, plasmids tend to be anywhere from thousands to hundreds of thousands of base pairs long, and they can replicate as the cell grows, and the replicated copies are usually distributed into each daughter cell when the cell divides. They have protein coding regions, which can include virulent determinants, and they can also carry coding sequences for antibiotic resistance, conjugation pili, or even entire metabolic pathways. Now, there are different types of natural bacterial plasmids, and these include resistant plasmids, which can carry antibiotic resistance. There's fertility plasmid, which, like resistant plasmid, can encode the conjugation apparatus, but these can move the chromosome. Some plasmids are coplasmid, coding for bacteriocin, which is a type of toxin that can kill other bacteria. There's also virulent plasmid, which code for proteins involved in resisting host defenses or also producing toxins. And there are a type of metabolic capacity plasmids, which code for proteins involved in certain metabolic pathways like nitrogen fixation or even lactose metabolism. So sometimes bacteria can transfer plasmid to another bacterial cell through conjugation, which is what we'll talk about next. So conjugation or bacterial conjugation is a type of mating between bacterial cells that requires cell-to-cell contact through a pilus. The mechanism of conjugation itself is a plasmid encoding mechanism that can mediate DNA transfer between unrelated cells. And these are called conjugative plasmids because they can use this mechanism to transfer copies of themselves and the genes they encode to new host cells. So conjugation requires a donor cell, which contains the conjugated plasmid, and a recipient cell, which does not contain the plasmid. Other genetic elements that can be transferred through conjugation, of course, include plasmid, but also the host chromosome itself, as well as other genetic elements that are unable to transfer themselves. So as we see here in this figure, we see on the left, we see a donor cell which contains the fertility plasmid represented by this F plasmid. And we call this donor cell an F positive cell or an F plus cell. We call the recipient cell that does not contain the F plasmid the F negative cell or F minus cell. In the first step, direct contact is made to the recipient cell via the pilus. And once direct contact has been made, the pilus retracts to bring the cells in closer. And then the cell pairs will stabilize and transfer will occur once the F plasmid has been nicked in one strand. One strand is transferred from the F positive cell to the F negative cell, and simultaneously the F plasmid replicates in the F positive cell. And next, the complementary strand is synthesized in the recipient cell. And once that is completed, this completes the DNA transfer and synthesis, and the cells will separate. And so the recipient cell is now also an F positive cell. So that is how bacterial cells can transfer plasmids that are separate from the chromosome. We're getting close to wrapping things up, but there's just two more things that I want to mention. The first is I want to mention how chromosomal DNA can be transferred by conjugation. So usually we often think about plasmids as being separated from the chromosome, but plasmids can integrate into the chromosome. And we call cells with an F plasmid integrated into the chromosome HFR strands or HFR cells, which stands for high frequency of recombination. And this refers to the high rate of genetic recombination between genes on the donor and recipient chromosomes. A high frequency of recombination cell is usually formed when homologous recombination occurs between an insertion sequence on the F plasmid and a corresponding insertion sequence on the chromosome, as shown here in this figure. So we see an insertion sequence here represented by the yellow box on the plasmid, which is represented in purple, and then also in a corresponding insertion sequence here on the chromosome. And so as a result, the F plasmid can become integrated into the host chromosome, which we see here. Let me zoom out real quick, which we see here, and I'll zoom back in. So you can see an example of what this looks like. The host chromosome is represented by green, and the F plasmid is represented by purple. And you can actually see an origin of transfer or ORET site here in the center, which is going to be important for transfer. So moving on to part 2, this shows how chromosomal genes can be transferred by an HFR strain. The HFR chromosome breaks at the origin of transfer within the integrated F plasmid, and this is the point where the transfer of DNA to the recipient begins. And this part 3, shows how this process occurs through conjugation. And the process is essentially the same for how plasmids are transferred by conjugation. However, the recipient cell remains an F negative cell and receives a linear fragment of donor chromosome attached to part of the F plasmid. And so in order for the donor DNA to survive, it must recombine with the recipient chromosome after transfer. So like transformation and transduction, genetic recombination between HFR genes and F negative genes involve homologous recombination in the recipient cell. So we covered a lot of information today, but one of the last things that I want to briefly discuss is how horizontal gene transfer can shape the genome. Recall that the accessory genome contains genes that may not be present within all strains of a group of bacterial species, but found in some individual ones. Horizontal gene transfer can shape the accessory genome as it is one of the primary mechanisms in which genes found in the accessory genome can be acquired. These genes can be related to characteristics like virulence, resistance, or even metabolic pathways. These also include genomic islands or pathogenicity islands, which have been picked up through the exchange of DNA. And genomic islands are a bacterial chromosome region of foreign origin that contains clustered genes for some extra property like virulence or symbiosis. And pathogenicity islands are essentially the same but contain clustered genes for virulence. The core genome, on the other hand, is the set of genes present in all strains of a bacterial species. Although horizontal gene transfer may be less impactful in the core genome, occasionally core genes, genes that are essential to survival and growth, can be acquired through horizontal gene transfer. Lastly, the pangenome is the complete set of the orthologous and unique genes found within a bacterial species. It is composed by the core genome, the accessory genome, and species or strain-specific genes. Horizontal gene transfer contributes to the diversity of the pan and accessory genomes. In some cases, genes acquired through horizontal gene transfer can expand the pangenome with the increase of the addition of new genes from other bacteria, thereby contributing to a more open pangenome. So next time we're going to do an activity where we take what we've learned in bacterial genomics part 1, 2, and 3 and apply how this knowledge could be useful to outbreak scenarios. And I'll send out an announcement ahead of time for the date of this session. So with that, I conclude today's lecture. There are some great resources out there that cover classical bacterial genetics, and two of my favorite texts that I used today are Rock's Biology of Microorganisms and Molecular Genetics of Bacteria. And I also want to give a shout out to my old mentor, Dr. James Graham, for allowing me to use some of his content that I came across in grad school. With that, I will stop the recording and we can move on to questions.